Hello and welcome to BioCode. In this video, we will see how we can create gene structure through gene structure display servers. Gene structure visualization is usually done for genome-wide association analysis because there are various reasons why we need to visualize structures, that gene structures. For example, when we have to compare that structure with other organism or other species. For example, we need to compare coronavirus gene structures with coronavirus 1 rather than the 2, so that we can see where the CDS is present, where the exons are present, and to have a better visualized way of biological data. To actually utilize gene structure display server, you need 3 or 4 types of formats and data format. For example, you can use bed file format, you can use gene accession number. For example, you can just input the accession numbers from NCBI, or you can use GTF or GFF file format, or you can put in sequence. But when you have to put in the sequence, that should be CDS sequence, and that should have a corresponding genomic sequence as well, so that it can know where the CDS is, where the exon is, where the intron is. But what at BioCode we recommend using GTF or bed file format or GenBank accession in most cases. We don't really recommend using sequence file formats because there can be multiple issues with putting in sequence. If you don't have a choice, then only you should sequence as first format. So let's really get started with our analysis. We have really, in our case, we have a GTF file format of coronavirus 2 that is actually producing COVID disease. So we need to create a gene structure display through gene structure display server, and all we really have to do is go into this link, and then we really need to select the format that we need to input. As in our case, it should be GTF because we have a GTF file format. Then all we really have to do is choose file, and once we click on choose file, then we really have to go into where the actual file is saved. In our case, it is saved on desktop, and all we really have to do is just click on submit. If you have a phylogenetic tree as phylogenetic order or you would like the output to be ordered, you can select that option from over here, and you can select the output as well. So that really depends upon your sort of analysis. You should put in that the tree, and that the phylogenetic tree in a numeric format over here, and it will sort the genes in order, in the order that the phylogenetic tree